Okay, thank you very much, everyone, for joining us at the first of the BSGCT Translational Seminar Series, which we are running in association with LifeArc and Catapult. Thank you for joining us today. We're going to be going through a broad overview of the developmental pathway of gene and cell therapies. This is the first of a series of 4 seminars. We're going to be going deeper into the subject over the next couple of sessions, and we'll be giving you more details about those at the end of this session and in your email. Please feel free to put questions in the chat. You just need to sign up into YouTube, put them in there, and they will be publicly viewable, and I'll be moderating the question and answer session at the end. So we're joined today by our presenters, Joanna Davidge and Kieran Hudson from LifeArc's partnership team, and they're going to take it from here. Thanks very much, guys.
Thank you, Alex. It's great to be here, taking you through this first webinar in the series. I'll get the slides up and we can get going. So this first webinar in the series, which we're hosting here at LifeArc, is going to give sort of overview of the development path and a couple of other aspects that are key to overall development and translation of cell and gene therapies, and then there'll be more detail provided coming up in webinars 2 and 3, and then hopefully we'll have a good case study or a couple of examples for you in webinar 4. So just by way of introduction, I'm Kieran Hudson. I'm a business manager in the partnerships team, where my responsibilities include management of the funding we provide to academics, particularly in the advanced therapies space, and Joanna Davidge, a principal business manager in our team, is also on the call. I'm going to take you through the slides, which should take about 20 to 25 minutes, and then we'll both be here at the end to answer some questions. So please do add them in the chat or the comment section as you go along. Alex will be collecting them and then asking them at the end. So do ask us anything, and we'll aim to stick around for a few minutes afterwards to answer anything you have. So what am I going to cover? There's going to be 4 main parts to this presentation. So first, I'm just going to give some information about LifeArc's support for advanced therapies and its role in the translational ecosystem. So if you're not familiar with LifeArc, or even if you are, more information on what it is exactly that we do, particularly relevant to advanced therapies. And then I'll get into the kind of meat of the presentation, a general overview of the development path for cell and gene therapies, an introduction to the key steps and aspects of it, and introducing some of the terminology that you may not be familiar with that will come up through the latter webinars. I'm then going to talk a bit about who the key members are for your cell and gene therapy development team. You can sort of learn who is needed to contribute to a successful program, and maybe look around and identify any gaps in your team. And we're going to have some tips on where you can find colleagues and team members as well. And then we'll end with just a bit of information about how academic development of cell and gene therapies, and particularly translational research, can be funded, and again, some tips for how to get to a successful application as quickly as possible. So LifeArc's role in the translational ecosystem. So LifeArc is an independent British medical research charity. We're funded through our own translational activities, and we have an aim to invest up to £1.3 billion by 2030 into translational research. Our capabilities and work falls into 3 main streams. So we have a translational science, we have our own internal science teams based at 3 locations in the UK. It's not really the scope of this talk. We provide translational advice. So that is to many partners, including government organizations, charities, and other partners. And we also provide translational funding. So academic funding in various areas. I'm going to talk a bit more about that at the end. And it tends to be a 2-for-1 package with us. So we also work very closely with the scientists we fund to provide translational advice as well, and to help shape the progress of their research. Just to highlight a few areas. So we have a philanthropic fund for rare disease funding, which has been going now for several years and is well established. We've awarded over 50 grants in various areas of rare diseases, and this now forms part of our recently launched Translational Challenge in rare disease, which you can find more information about on our website. And specifically for gene therapy, so a common theme for LifeArc really is bridging the gap between academic and commercial research, and particularly identifying bottlenecks and areas where we can deploy our resources to help overcome roadblocks. So in advanced therapies, an example is often a major challenge in the translation of academic gene therapy research is the cost of getting sufficient clinical grade material for clinical trials. And we've sort of identified that and taken action over the recent several years to try and address this through a couple of initiatives. Firstly, we've partnered with the MRC and BBSRC to create a network of innovation hubs for gene therapies, and this provides clinical grade products, facilities, and translational expertise to academics. So I'd encourage you to look into that if you're not familiar. The hubs are in 3 locations across the UK currently, so King's College in London, NHSBT in Bristol, and at the University of Sheffield. In addition to this, LifeArc has committed to a £5 million annual fund, the Gene Therapy Innovation Fund, which I co-manage along with Joanna, and we'll be discussing a bit more about that in the funding section of the presentation as well. So, our overview of the development path for cell and gene therapies. So this is kind of a introductory, simplified, high-level depiction of the path. It's more complex than this in reality, and the subsequent webinars will go a bit more into detail, but this is just setting out the sort of overall definition of the different steps and some of the key aspects of them so that you can see the sort of path in a holistic view, which I think is going to be a repeating theme in this webinar. So broken it down into 4 different categories of activity. The sort of earliest that will begin typically is non-clinical discovery. So this is initial proof of concept and validation work. What is being discovered is your new potential therapeutic product. The second stage then, which builds off this once you have a candidate product, is non-clinical development. So this step is all the work done to acquire data in the lab to enable clinical trials. Initially building towards the data package needed to get your first in-human clinical trial, but non-clinical development work actually continues alongside clinical development and contributes to further clinical work and the authorization and adoption as well beyond the clinic. So as I say, this will be explained more in subsequent webinars, but we're going to give an overview here. The next stage, once you've initiated your first in-human trial, is clinical development. So this is all the work done in patients and humans, and the work that contributes towards your eventually getting marketing authorization, having your product approved and adopted and into patients more broadly. And then there is obviously, although reaching your first marketing authorization is a huge milestone, there is a lot more work that gets done after that. It's really beyond the scope of this webinar and mostly usually beyond the scope of academic research, but it is something that you always need to be aware of. And as I say, the holistic view is really a theme here, and maintaining that view on how you get to clinical trials and beyond throughout the process, right from the first step. So the first step of development, once you've got your thesis and your proposed therapeutic approach, is non-clinical discovery. So this work is to demonstrate sufficient proof of concept for your therapy and your proposed treatment approach to justify the next steps, the non-clinical development and further development steps. The information that you gather here will also direct the non-clinical development studies. And yeah, so the typical entry point here is generally once from your sort of ongoing underpinning research, you have a thesis for a therapeutic approach and you want to take it forward and develop it. So the key objectives here are to understand the disease mechanism, ensure that your proposed approach is actually going to work to treat the disease in suitable model systems, demonstrate sufficient efficacy so that your therapeutic will have the effect you desire, which you propose will treat patients. It's not a full non-clinical development data package, so you won't get the full efficacy data, but you need enough confidence and proof of concept to take it to that next stage. You also need some evidence that the treatment is likely to be safe. So is your proposed mechanism of treating the disease likely to be safe and tolerated by patients? And at this first stage, you should really start to develop a TPP, or a target product profile. So this is a summary of the desired characteristics of your proposed therapy in the disease you propose to treat. So you're already at this earliest stage thinking about what that sort of end of the pathway is. What does the product that you will eventually develop look like? How you'll use it to treat patients, and that should be guiding every step of your research. So a few key considerations for this step. So make sure that whatever models you're using at this stage are appropriate and that they truly relate to the disease as it biologically happens in patients. I'd say appropriate, and really you want to find the best models you can. This is something that's always a key question that funders ask is, are the disease models the best ones? Are there better ones out there? Do you have access to the appropriate ones? Be sure at this stage that you understand the unmet clinical need, and this can be through talking to patients and clinicians. There'll be more about that in the team section of the webinar, but you really need to understand what patients need, what the standard of care is out there, and what you need to do to be able to improve on that. So there'll be a huge difference between an ultra rare disease that is largely fatal in early life versus a chronic, broadly suffered, reasonably well treated disease where you might need to have quite a different product to actually make an impact and a treatment that's going to be adopted and make a difference. Definitely try and tap into pre-existing data where available. So if there's natural history data, assembling your rationale for why your treatment's going to work, why it's going to be safe, look for what's out there in terms of real world data and previous studies. Think about intellectual property and freedom to operate. So in advanced therapies, cell and gene therapy, it's very likely that you're going to be using proprietary tools, proprietary methods, perhaps manufacturing processes, and you need to make sure you have access to those for every stage of development. You might have the rights to do your academic research, but you need to be thinking about rights for your clinical work and even rights for the downstream commercialization and translation into patients, and have confidence that you can get the rights down the line. This is often something that comes up in proposals and isn't fully explained. So it's something you need to think about to make sure, again, looking at the whole pathway, that you can make it all the way there. And also just be aware of what your options are for funding the further development. The development starts to get expensive. There are schemes out there that will typically have quite specific requirements of what they need in order to secure funding. So you need to make sure that the work you're doing will give you everything you need to take that next step. So the non-clinical development, this is where you're gathering non-clinical data to support your clinical trial applications and further steps. So the sort of entry point for this is basically where you have your candidate product and therapeutic approach. You've settled on it with your proof of concept data, and you're now gathering all the data you'll need for your regulatory approvals. You've probably also at this point secured some translational funding. This is generally beyond what sort of typical fundamental research funding will cover. So the objectives here, to make sure you have robust analytical processes that allow you to characterize your product. So this is about being able to demonstrate the product is what you think it is, that it's consistent, that what you put into, you're suggesting to put into patients, you're well aware of everything that's in there, and you can consistently say that you're producing the same batch every time. And that is the batch that you need to be doing, the product that you need to be doing your studies on. So you'll complete safety and toxicity studies, so gather evidence in animal models that your product is likely to be safe and toxic enough to convince regulators that it's appropriate to advance to clinical development. Get biodistribution and pharmacokinetics data. This may be from new studies or for advanced therapies if there are precedents out there. Regulators tend to be quite pragmatic about what is needed, and they will also consider real-world data if your product is comparable to something that's already been through trials or been approved. Biodistribution is looking at where does the product go in the body and what does it do when it gets there. Pharmacokinetics, the effect of the product in the body sort of over time until it's cleared. So this can also be linked to your rationale. Is it a product that is only active for a short period of time and that's all you need? Do you need something that is durable for months or even years? Is it a cell therapy product that will expand in the body? Is it a single dose? Things like that you can link to your rationale and your justification. And studies in multiple animal species at this point may be necessary. This is probably a difference from the more traditional small molecule and biologics development pathway, where 2 animal species is fairly typical. Here it might depend more on what appropriate animal models are available and what comparable data there is. So this is something where you need to have a good rationale and again be able to justify your approach. So key considerations: engage with the regulators early to obtain scientific advice. They are the ones who will be deciding on your clinical trials application, so you need to be confident that what you're gathering will be sufficient to convince them. And you can speak to them about this, and we always advise to engage as early as possible. Particularly now, there's quite big delays in getting scientific advice from the MHRA, so get in the queue as soon as you can. Manufacture the product using the final product production process where possible. So you need to have the data around the product that you're actually going to put into patients and ensure that you're, again, having robust analytical processes to show that what you're putting in, you fully understand. So the earlier you get your final manufacturing process set up, the better. Otherwise, you might have to do studies and further experiments to show that what you're producing in the end is equivalent to what you did your studies on, which can make things a lot more complex. And for these non-clinical development studies, they mostly need to follow GLP, good laboratory practice. So that's typically beyond the scope of what universities and academic labs can do. So most often this is done in a fully accredited contract research organization lab. So you need to think about who's going to be doing these studies to this standard and make sure they have all the right processes in place. And this is one of the sections where there's more info going to be coming up in the subsequent webinars. So clinical data development. So once you have your sufficient data for a clinical trials application, it's been accepted, you enter clinical development with your first in human studies. You need your site or sites lined up and your clinical protocol agreed. The objective here is to demonstrate your safety and efficacy in humans, working towards your marketing authorization. So objectives: you need to make sure you can manufacture sufficient GMP, good manufacturing process grade product. So that's a fairly universal standard of what is required to manufacture a product that's going to go into clinical development. And you need to have that sort of in hand, or at least a process in place to be able to access it throughout your clinical development plan. And there are 2 main aspects to sort of early clinical development. What is traditionally classified as a phase 1 study in terms of determining just the safety of your product and identifying the effective dose through single or dose escalation studies, and then demonstrating the efficacy, which would be more like a phase 2A type study. This is again a difference for advanced therapies versus the more traditional small molecule or biologics model. Often phase 1 and 2A studies are combined, and you also won't do phase 1 in healthy volunteers for numerous reasons. You typically will go straight into your target population and then escalate and include expansion steps for your efficacy. So there's again, the regulators tend to be very pragmatic about this and will discuss approaches and accept a reasonable justification, but you need a good plan for how your clinical development is going to work, and you're going to get the evidence you need to demonstrate your safety and efficacy. So key considerations: this stage is very expensive compared to a lot of academic research, and this is where you really need specialist funding. There are funders that can cover early clinical development work, and we'll talk about that, but a lot of this is typically borne by commercial partners too. You need to make sure that you have a manufacturer lined up. These products are complex, and it can be there's not a huge list of companies out there that can do it, so you need to make sure you have someone that can make it for you. We'll talk a bit more about that as well. With your trial design, as I've said, regulators take a pragmatic approach, so consider precedents, look at what other people are doing, and just come up with a good rationale. And you need to be following good clinical practice for these clinical trials and make sure that whoever you're working with to run the trials does so. And again, there's more info coming up in the later webinars. And then beyond clinical trials, as I say, is really beyond the scope of this webinar, but it's something you need to be aware of. You need to know how your product is eventually going to make it to patients and be commercialized. Some key considerations, there needs to be a plausible route to the product reaching the market. The sort of simplest level is thinking about whether you're licensing to an existing company. Are you potentially building a package that's enough to justify a new spin-out company, and a good rationale for the approach you're going to take, and then what is required, awareness of what is required for adoption of the therapy by the health service, and how you're going to meet those requirements. So that's the overview of how you get there, and now the question is who do you need in your team to be able to get there effectively? So we've identified 5 kind of key areas where you'll find you need input and expertise, but this is by no means the full list, and there are other sources of expertise and aspects of expertise that you'll probably need access to as well. So I'm just going to run through each of these, and then also just a few ideas about where you might be able to find the expertise if currently it's not something that you have yet in your team. So the translational scientists, I'm assuming most but not all of the people on this webinar are probably scientists. So their role generally is to do most of that non-clinical discovery and a certain amount of the non-clinical development work. So they come in with the understanding of the disease mechanism. They have a thesis about their therapeutic approach and product. They select their candidate to take forward into study and develop and test the different versions to choose which one to take forward. They understand the disease models, and they select and bring in the appropriate one to their lab, and then generate most of the non-clinical evidence in vitro and in vivo. So this is where the sort of classic science is done. So universities, government research institutes, charity research centers all do translational science. Industry obviously as well does a lot, from small biotechs right through to big pharma. There's a lot of translational scientific expertise in industry as well. So in terms of building out the team, so you need expertise in the patients, which can come from clinicians and or from patient groups. So someone who understands the target indication, the disease you're trying to treat, and the size of the affected population. They know what the standard of care is, so they know where the unmet need is. They know what the requirements are for a new treatment to really make a difference, and what the sort of limits are in terms of what patients will accept based on what their quality of life is like and the changes that are possible. If, for example, you're proposing quite an intrusive method of delivery, or lots of repeated dosing, whether that's something that they will tolerate, or the alternatives they have might make that less tolerable. And then obviously they can also provide access when you're ready to take that step into clinical trials to help with recruitment. And they'll help you shape that target product profile through this combination of understanding. So as I pointed out in the discovery stage, you really already need to be thinking about your target product profile. So you want to be engaging with these clinicians early. So you can find clinicians in hospitals. You can reach out to disease-specific charities and the disease you're interested in, and patient groups as well can provide you access to the information in the patients. Manufacturers, so you need someone to make the material for you at the right grade. Particularly valuable to find someone who has experience in the specific product and technology you're using. They'll be responsible for meeting the regulatory requirements for the product. You need to ensure they have capacity, which can be limited. So again, talk early and know who you're going to have lined up, and make sure they have manufacturing slots available for you when you need them. But you also need to think about a route to commercial scale. So how do you go from the sort of early studies you're doing through to larger studies and full-on adoption? Does the manufacturer have capacity to expand? And if they don't, what is the route? Is there a route? Would they be able to license to another manufacturer with similar capabilities, or transfer the technology somewhere else? Something to think about, so you don't sort of get stuck down the line where you've got an approved process and product, but no way to scale it up. So a lot of this work is done by contract development and manufacturing organizations. Some universities do have some facilities for early studies and small scale. I've already mentioned the innovation hubs for gene therapies, and some of the catapults can help with this as well in terms of expertise. So you'll need someone with expertise in the regulatory side, understanding of the regulatory requirements. They'll help you assemble and ensure you have the right data. They'll also be responsible for CMC, so kind of understanding the manufacturing process, the product characteristics and required product testing you need for approval and authorization. I've mentioned the MHRA you can go direct to for advice, but given the timeframes there, you may also benefit from independent consultants, many of whom are ex-regulator or ex-industry. Some universities do have internal translational support as well, and funders such as LifeArc can also make connections to the right people. Contract research organizations do have their own expertise as well, and again, people like the Cell and Gene Therapy Catapult can be a good source of regulatory advice as well. So there's lots of people out there where you can make the right connections. You need someone who understands the IP and ideally the business as well. So the intellectual property environment and freedom to operate. They can help you secure licenses you need to any background IP and help you with the early kind of commercial approach and business model. So University Tech Transfer Office should generally be your first port of call with this. They tend to have a lot of experience with therapeutics development, and they're usually very interested to hear about therapeutics opportunities. So do reach out to them early and make sure that they're fully aware of your project in the background. Some universities now have entrepreneurs in residence, so successful entrepreneurs that come and talk to academics with business who can bring their business experience and talk about business models and approaches. And you can also go to external consultants. And to say that list is sort of the core team normally, but it's not exhaustive. You may need access to experts in statistics to help with animal studies and trial design. There are clinical trial units around the country that can help with designing early phase clinical trials and may even be able to host some of the first in human trials. Health economics is always an important area to ensure that the trials collect proper economic and quality of life data to help profile the value of the therapy. This is somewhere else where the Cell and Gene Therapy Catapult, for example, can help. And just a final few slides about how to fund the translational research. So what's out there beyond sort of the traditional research council and other funding for underpinning research. So I'm going to speak about the MRC and LifeArc funding in a bit more detail on the next slide. But just this kind of shows that there is sort of a sequence in the Medical Research Council funding options through early discovery into non-clinical and clinical development through a number of schemes. LifeArc's philanthropic fund and then other funding for academics tends to be in this area from sort of non-clinical discovery through to mid-clinical development phase. But we're always happy to talk and provide advice on where your project is and what the appropriate stage to engage with us is. And we also have the Gene Therapy Innovation Fund, which I'll talk more about. There are other sources, so charities, other charities, disease-specific charities do often have translational funding. So I picked out a few larger examples, the British Heart Foundation and Cancer Research UK. If that's the area you're working in, they will probably have translational funding. Smaller charities often do as well, that covers non-clinical work and also clinical work. And if you have industry partners, there are sources that focus primarily on funding industry. So Innovate UK is part of UKRI that provides funding to industry, but often in collaboration with academics. And they have a biomedical catalyst, which is actually a specific program for life science, but you can also get more of their general funds as well for appropriate programs. So just to pick up on a few of those key funding schemes. So the first step in translation is typically proof of concept funding. So MRC Impact Accelerator Awards are awarded to universities. They typically fund sort of small pots of cash for projects to get proof of concept data. They're typically quite light touch applications and quick turnarounds. Some universities have other schemes with their own funding as well, maybe run through the tech transfer offices. As you develop, there's a new recently launched scheme from the MRC, the Development Pathway Gap Fund, which kind of bridges the gap between Impact Accelerator Awards and the next stage. And that's for slightly larger pots of money for slightly longer projects to get more proof of concept and evidence that will then set you up for the kind of flagship MRC translational funding, the development pathway funding scheme. This funds everything through late stage non-clinical development through to early clinical research, and there's up to 30 million available. There's a regular rounds, and yeah, there's quite substantial amounts of money available for that once your research is in the right stage. LifeArc has various funding schemes. The philanthropic fund is kind of the constant rolling scheme for rare disease indications, which can and often does include cell and gene therapies, with up to 1 million available per project. And there's the specific gene therapy innovation fund, which specifically covers the cost of manufacture of GMP product at one of the innovation hubs. There's also 5 million available per year for that, and it's not capped per project, so they can be bigger grants as well. But it is intended to supplement other funding, which covers the translational aspects. The GTIF only covers the manufacture. But I'd encourage you to reach out to us if you have questions about LifeArc funding, because we're always happy to discuss your research and where the opportunities are. Just my final slide, just a few top tips for translational funding and maybe differences to other funding applications you're used to. So you'll typically need a stage development plan, clear work packages, and quantified milestones that you'll achieve so that funding can be gated. As these are big grants, they tend to be tied to achieving certain milestones. And if you plan early that's how your research will be set out, that will make it a lot easier to get a good application in. Make sure you link the models you have to the actual clinical outcomes and fully explain what you expect in the model versus the clinical outcome. This is something that comes up quite often in our reviews and panel discussions. Make sure you have a full target product profile and you know what you're really working to from that early stage, so that you can rationalize all the experiments and data you're seeking versus your final outcome for patients. I'd say work with your tech transfer office as soon as possible, and then provide them with as much information as you can on the IP and any processes you're using, so that they can provide a full plan in your application of any issues related to IP and how that will be mitigated to ensure that your product can make it out there onto the market. And yeah, make sure you do have some idea of how your product at least could make it to market, and include this, because that's something, again, that funders will look for. Is there a rational, feasible plan for how your product will get onto the market? Because they want to make sure that the research they fund has the best chance of reaching patients. So that's it. Here are the contact details for myself and Joanna. As I say, especially if you have questions about LifeArc's funding, do reach out to us. We'll be always happy to receive your emails and have a conversation. So yeah, and I'm just going to say thank you and hand back to Alex to answer any questions.
Thank you very much, Kieran. You squeezed in an enormous amount of information into a really tight time slot there, so I really appreciate it. Yeah, I think it was really important what you said about maintaining that holistic perspective, that end-to-end perspective. So it's, I think the emphasis there was very clear. It's you need to make sure that you've got that long-term view even before you necessarily have your final piece. So I'd also like to thank everyone who sent through questions ahead of the seminar today as well. So I'm going to open up with one of those. Kieran, you touched lightly on IP and how important it is. We have a question from Eleanor at UCL who wants to know, when is it appropriate to start meeting investors or venture capital funding, and is that before or after securing your IP?
So I'd say, well, I should caveat that in my sort of other life, I'm a tech transfer officer. So I come with that perspective on IP management. I would say you want to sort of be securing it as early as you can, that you have to, but certainly before you talk to investors. I think that's one of their first questions they're going to ask in the diligence is, what's your IP position? I would say take the advice of your tech transfer officer, but I'm kind of obliged to say that. But yeah, make sure you're at least aware of your position. They're going to be asking those questions, and doing the diligence is one of the things they care about the most.
Thank you. And we had another question in the chat from Stephanie, I think this one will also be covered partly by our next seminar, which is on regulatory considerations. But if either of you would like to weigh in now, when are you hoping to use existing, such as real world and previous trial data, to support a new trial? How similar do the products need to be?
So I can take that one. So I think the more data that is coming out for certain capsids in humans, the more the regulators are prepared to use that data to mitigate the path for the next therapy coming along. So just to use an example of the dopamine transporter deficiency project. I don't know if there's anyone in the audience there who developed that. But because a trial had gone into the clinic for AADC using the same AAV capsid and the same route of administration, the regulators didn't then see a need for repeated large animal biodistribution for that next program that followed on. So that was a really clear example of an expedited path to clinic where real world data had been used from a forerunner. And we've got, I don't know if we've got Joe on the line who is working on a tyrosine hydroxylase program, which is, we're hoping, going to be the next in that sort of pipeline. And also we'll hopefully have an expedited path to clinic. So, I mean, the regulators are looking for you to go to them and looking for you to suggest these paths based on data that you know is out there. So, you know, in that case, we went to them and we suggested that we didn't need to do any GLP work. We presented the case and they agreed with us. So, you know, that's an example of where they've been pragmatic. There isn't really a well-trodden path like there is for small molecules. So they're looking to you to really suggest those routes based on information that you have.
Makes a lot of sense. So yeah, unlike in some other more well-trodden modalities, it sounds like it's much more negotiation with yourself and the regulator and a discussion. So I think we started talking about clinical trials there as well. We have a question from Danielle at UCL. What are the roles and opportunities for medical doctors in facilitating the translation of this kind of science?
I mean, a clear role there is an understanding of the disease and of the patient population. I mean, we absolutely always will suggest to basic scientists to link in with a clinician right at the beginning. You are the guys who understand what the disease looks like, what your patient population looks like, and what a potential therapy needs to address. So, I mean, absolutely a key role right from the start.
And from Manojat Iminil, are there any developments or guidelines for shorter or expedited clinical trials?
That's a tricky one. I think it's case by case. We're obviously looking, as Kieran said, it's not a path like it is for small molecules. So often that first in human trial is also looking to, you know, evaluate efficacy. So yeah, it really depends on the endpoints. Pick out clear endpoints to show efficacy if you can, and always speak to the regulator early on about your trial designs. I think your CTU could also probably input there.
Thanks very much. Yeah, I should also point out that a lot of the questions that we're receiving are on regulatory concerns, which will be the whole topic of the next seminar, and we'll really focus down into that one. A question from Sarah as well was asked about PPI inclusion in the early stages of preclinical research. So that's patient and public involvement, I imagine. Any comments on where that's appropriate and how that looks?
Yeah, I mean, that's another area where we're really trying to drill down into at LifeArc. It's not something that I think we're doing sufficiently right now, and we are working on that. So we're actually recruiting a patient and public engagement lead to really help us get the best out of that. Also in the philanthropic funds, we're looking to introduce a larger PPI&E section, because obviously you need to speak to patients and understand feasibility. You need to understand what the endpoints are that are important to them. You know, we've heard horror stories of therapies getting to HTA where they haven't yet engaged with patients. And actually when NICE goes to do that HTA analysis, they discover that the endpoints that this therapy is looking to treat are not those that are the most important to patients. So I don't think we can stress this enough. Engage with your patients and patient groups, you know, as early as possible, right at the beginning when you're thinking about your target product profile.
This potentially is another role for medical doctors as well. They usually have good connections with these patient groups, so it's really crucial for scientists to engage with doctors and the patients. And it comes back to Kieran's point about maintaining that holistic perspective, and that applies at every stage of this development process. We'll be discussing a bit more about some of the terms that are coming up in this seminar in follow-ups as well. So our next seminar will be on regulatory considerations, and that's going to be on the 25th of October. So that's next Wednesday at 3 p.m. Just like this seminar, it's going to be recorded as well and will be available on our YouTube channel afterwards. Please do feel free to reach out ahead of time if there are any questions that come to mind, or if there's anything you feel we haven't covered today. And next seminar will be presented by Catapult. There will then be 2 more seminars following that on non-clinical aspects of development. So what happens in that package that Kieran was discussing today, and then looking at a case study to describe people who have actually been through this process. Thank you very much for joining us for this seminar today, and we hope we'll see you on the next one.